Great. Hello, everyone. Welcome. We are excited for you to join us for today's webinar. So today we'll be talking about building an AI-ready foundation for antibody discovery. My name is Monica. I'm the head of go-to-market at MiLaboratories, and I will be your host today. We're also joined by Sam Poslavsky, who is the CEO of MiLabs. Just a reminder to everyone, the session will be recorded and we'll send it out tomorrow. And you can also drop your questions or comments directly in the Q&A, and we will answer them at the end. As a brief intro on why we're doing this webinar, we host a High Affinity Talks webinar series where we bring in researchers in the antibody space. And 2 weeks ago, we sat down with Andrew Bradbury, who talked about his benchmark AI competition. And essentially, the results didn't live up to the hype. AI was worse than random picking for all but one algorithm. And the highest affinity AI design still failed basic developability checks. And that's why we're doing this webinar today, to talk about how to build the structure and the data layer. So there's a foundation that AI can actually build on, not just more data, but the right data and structured so that the models have an actual chance at working. In addition, we'll also cover each step from raw sequencing data to lead selection and talk about computationally what each step needs on record so you have clean, structured, and reproducible data for this foundation. So let's get started. And first, I will pass it over to Stan, who will introduce us and the agenda for today.
Thank you very much, Monica. So, yeah, this is a practical computational workflows to find and de-risk antibody leads and build the AI-ready foundation. First of all, just to make a clear statement, we're not AI skeptical. AI is very useful. It works, AI, machine learning, but the key is that the AI model is as good as the data that is fed to the AI. So that's where our focus today. While we're talking about this, Monica already mentioned that we hosted 5 talks with Andrew Bradbury, where we discussed this in a very deep biological details. But in general, a few words about us. We are developers of MiXCR, a tool that many of you know from the immune repertoire sequencing. So we work a lot with academia and industry, with different teams who run antibody discovery campaigns. Yeah. And today we want to share some of the learnings, some of the experience that we aggregated from many of our collaborators, especially in academia and industry, who are using AI and machine learning to build models and predict better antibodies. So the focus will be biological. So today we will be focusing on one specific type of experimental setup. It's for binder discovery. It's phage, or well, just in general display technologies. Yeah, where there are different types of experiments, of course. Yeah, and one of the most widely used approaches to generate the data foundation for machine learning and AI models is with display technologies, where you can quickly start from the high diversity library and then go through the panning rounds to find high quality leads, where the quality may be affinity, maybe specificity, maybe developability. Yeah, and these type of experiments is a workhorse for the modern AI to build the data. And one of the key bottlenecks, of course, here, that's what we're focusing today, is the analysis. So we are really, really good in generating the data, generating libraries, and doing all the biology with phage or yeast or whatever types of displays. But then the data analysis, structuring the data, making it useful for real, for AI and machine learning models is the bottleneck. So the key promise of AI, and by the way, when I'm saying AI, I'm actually I'm actually not. It's not only AI, but it's also just traditional machine learning, where we use machine learning models to find something and to do something. So AI, I'm using in a synonymous way with machine learning. Yeah, and the key promise of this modeling and AI is, of course, developability, affinity for lead selection. Yeah, generating good libraries in silico. And the key problem, let's say, the key bottleneck in this is the data. Yeah, one of the first obvious statements is garbage in, garbage out. So if we train our models on just a small set of antibodies, yeah, then the model will inherit that bias, that artifacts, and blind spots. Yeah, the data itself, as I mentioned, requires scale. So we need a lot of data to train good models, but at the same time, the data itself is not just a scale, but the structure of the data. Yeah, ability to reproduce, yeah, labeling the data, not just a spreadsheet with few bits. The structure of the data is a key ingredient for good modeling. Yeah, so, and the bottleneck is not the model itself, but the data foundation. So today the plan is the following. So we will be discussing the kind of what we are calling a cheat sheet, how to do a proper analysis of display experiment data to build a solid data layer, to go through the main caveats in the analysis of this data and what should be taken into account when building the data foundation. Yeah, the key, let's say, problems that we see some teams are doing, and then having problems in the model training. We'll also have some live demonstration how this may look like in the reality. Yeah, from row sequencing reads from the FASTQ files to the lead panel and to the data foundation that is used for the training, structured, labeled. Yeah, at the end, please ask questions. You have chat, you have Q&A session. And basically, we will learn how to structure the data on different levels, on the level of the study, on the level of antibody clusters, on the level of individual antibodies, and how to integrate the wet lab loop to with this NGS data to have a really robust data set for training. So, what makes data AI ready? Few things. Yeah, the model training is the process which we run on many, which ideally should be run on many discovery campaigns on many different types of data, and reproducibility here is very important, because if we train if we have a model for, let's say, predicting developability or predicting, let's say, even affinity from the deep mutation scanning data, yeah, then if we run this with different versions of the software under the hood or different parameters, obviously this will not result in a stable model. Structured, so we need to structure the data in a way that is digestible by AI, clean with no PCR or sequencing biases or whatever can really destroy our data set for the model to train. We don't want to learn from the errors. We want to learn from the data that is clean. And labeling. Labeling is one of the key things. So for every data point, and with NGS, we are able to generate billions of data points or signals. We need to have a clear label, what it is, how this data point appeared, what it is, cluster or antibody or study level label, etc. So that's we will go in details in a few minutes. Okay. We are working this for quite some time and we have an open source and transparent environment. That is, many of the algorithms and many of the ideas that I will be showing, they are available on GitHub, and very transparent white box. So this is part of our Platforma ecosystem, which provides the whole layer of data structure going from the raw sequences to the lead selection and building this data layer for model training. Monica, I want
Yep,
to,
I'm going to
yeah,
launch the
yeah, yeah,
first
yeah, yeah.
poll. So while Stan is going to the next slide, you can, oh, sorry, not that one. I just launched a poll, and so yeah, which we're curious to learn which software are you using
Mhm.
for antibody discovery?
Yeah, and whatever software you're using for antibody discovery to build a data layer for model training, we always go through the same steps from the data, from the raw sequence and data, annotation of this data, numbering, clonotyping, characterization on the level of the study, yeah, repertoire characterization, quality controls, clustering. Since we're talking about binder discovery with display experiments, it's a very common tool to antibody clustering, enrichment to track what are our enriched leads, developability assessment to assign easy, let's say, to score the liabilities that are easy to score based on the sequence, based on the structures, integrating of the assay data if that we have downstream, and lead selection. So basically, those are standard steps that we go with whatever we use for antibody discovery. So let's go one by one, starting with the repertoire characterization. When we are building data for AI or for us humans, we always need to start with repertoire characterization and label the data before we even consider individual antibodies. We need to understand overall how our study is good. With display experiments, there are two specific metrics that we want to look before we go into the details of individual antibodies, clusters, etc., etc. So one metric is rarefaction analysis. We want to see with whether we have enough diversity of the library that we expect, whether we have the funneling effect for the display experiment. We want to see that we have some dominant clusters of antibodies. Those are basic characterizations that we want to do to understand that the study is overall good or not, it's successful or not. If it's not successful, we're not going to use it for anyone. So the first thing that is a pretty simple but very useful instrument to understand overall whether the study is good and assign this, label to the study itself is a rarefaction instrument. So basically, the question that we are asking before we even consider the antibodies that are there inside the study: did we sequence to enough depths to capture all binders? Yeah. So I didn't say this assuming that this is an obvious thing, but the data layer is always about NGS. Yeah, and NGS gives us a scale. And when we do NGS, we sequence. Obviously, we cannot sequence to like 10 in the power of 12. But the question is whether we're sequencing to the depths enough to capture all signals required to understand that the study is good. And the rarefaction analysis gives us this instrument. So basically on the x-axis, we have the sequence and depth. On the y-axis, we have the estimated number of unique antibodies that we're getting. And if we look on rounds of enrichment in our library, we can see that we expect at least that the screening library of high diversity is not sequenced to the full depth. And if we sequence more, we get more diversity. And for example, on the left plot, you see an example from immune library. Of course, this will depend on what kind of library we are using for the screening, what kind of the display technology we're using for screening, etc. There are many factors. So a few examples here. Immune library on the left, the library itself, green, is growing. Very nice. So the more we sequence, the more diversity we get. That's what we expect. And round one and round two, they collapse, meaning that, so they are saturated, meaning that we sequenced to a full saturation. If we sequence more, we are not reconstructed more antibodies. That means that in our study, we captured all required diversity. If we have binders there, we sequence them. They are in the data. Yeah. The other examples that you can see, and this basically for the wet lab, that's very useful. That's like sometimes we need to decide how many rounds we should sequence. And for example, for high diversity synthetic library, like the middle one, yeah, we see that round one is not saturated compared to this immune library. Yeah, round one is still growing, round two is still growing, and only round three and round four we sequence to saturation. So it's saturated, so we're good here. Yeah, and in some edge cases and some experiments, which are using a more diverse libraries or for high diversity of like special types of displays. Yeah, we may not have saturation at all, and you see this example on the right. When we want to run it at a scale and generate many experiments, many studies with a lot of data points, with a lot of data points at scale in a reproducible way, we want to see always picture like left one or the middle one. Then we have everything sequence, the data is there. Yeah, and situation like this is not good for the training. But even if we have saturation, this saturation may be a result of just absent binders. If we don't have binders at all in the data, then what we see on the left, yeah, will be the same. With round 2 or round 3 or whatever, or round 1, we don't have binders, so there is no diversity. And when I will switch later to some live demonstration, I have a very good experiment to show how this can look like in different experiments. So to understand that we have binders, that we have expanded clones or enriched clones, enriched clusters, enriched motifs in the data, the second very useful metric that we found works really nicely is Gini index. Basically, this is just a estimator of the diversity that allows us to track the clonal dominance across the routes. And again, on the same example, so the left is the immune library, which already starts with, because it's immune library, yeah. So it's already starts with the high Gini index. So there is some expansion already. The repertoire is skewed, yeah. And then we're growing nicely to almost 1 in the round 2. This means we have real growth of antibodies. There are binders. Well, let's say there are antibodies. There are clusters of antibodies that are enriching across the rounds. Good. Here, same story. Yeah, it's good. And we are starting with lower Gini index, 0.4. So the library was really even, more or less, at the start. And then we progress to almost 1, meaning that, yes, now at the final round, we have all the information. We have all enriched clones, and we are confident that we can reconstruct the trajectories of those clones grown across the rounds, meaning that we have signal. And the last picture is there is some enrichment. There is enrichment, but it's not enough. So here, this point, 0.25, it's lower than this one, 0.4. So here, this example tells us that this is something that is like not unlikely we will have enough signal to confidently identify the signals. So the next step, if we are good here and we have this information on the level of study, we can proceed to the next step and classify data points on the level of individual clusters. So for display experiments with immune libraries or synthetic libraries like combinatorial libraries, antibody clustering is a very useful instrument. Just one remark. We are not using, obviously, antibody clustering for deep mutation scanning experiments, yeah? This is only for let's say, combinatorial libraries, rational library design, or immune libraries. And the idea is that with the sequencing, with NGS, we have sampling. So in the screening library, we never capture full diversity. And then across the rounds, we're not capturing the full diversity. So we want to cluster antibodies into groups or families, just clusters, to have a more reliable signal across the rounds. Yeah, enrichment signal. This is one goal, and the other goal is to actually use clustering information to diversify lead panel and diversify those antibodies that we are then using for wet lab validation. So 2 goals here for the clustering. Depending on the specific experimental setup, of the specific design, display technology, and initial screening library, yeah, we may use different methods for the sequencing. Today, in our experience, the most reliable and the most robust way to do sequence-based clustering is using MMCQS2. Yeah, so far it works best, especially on the scale when we have a lot of antibodies. There is one caveat. Bioinformatically, this tool is a very, very nice tool, but tuning this tool to work on your specific library with your specific diversity and specific structure of the library requires some bioinformatics tweaks. For example, for antibodies, the frameworks are fixed, yeah, and on the length of the full VG region, yeah, for the entire repertoire, if we have millions and millions of antibodies, they will have more or less same framework. So when we want to do clustering, yeah, for such type of libraries, if we don't have diversity in the frameworks, we want to cluster just based on the CDRs, yeah. And in general, for display technologies, for binder discovery, our recommendation and the number that works best across all the studies that we saw and did ourselves, 95% similarity on the protein sequence of the full VG region, or just CDR, CDR1, 2, 3, yeah, concatenated is the threshold that works well to reconstruct good and reliable signal in almost all experimental setups. Yeah, sometimes, and when we don't have good enough, let's say, funneling in our enrichment for certain reasons, there may be a very complex target, there may be like specifics of the biology that we're doing. Yeah, we can cluster just by the CDR3. Yeah, and this will give us, let's say, we will kind of zoom out from the repertoire and have a better signal on the level of bigger families. That will result in the fact that it will be harder to identify individual antibodies within those clusters. But that's the trait. And if we're doing some optimization experiments when we don't have big diversity, we may even use more stringent criteria to do, let's say, what we call a variant resolution and go deeper into the level of individual, let's say, variants. There are two other types of clustering that are useful, and I will briefly cover them on the live part, is paratope-based clustering. Sometimes we don't have enough diversity, yeah, in the set of clusters or antibodies that are enriched across the rounds. So if we want to diversify a panel more and see how diverse this panel on the level of its function, yeah, on the level of paratopes, we may use paratope-based clustering, and Parapret is the best tool that so far we discovered works nicely for the clustering, not for the predicting the paratopes. And downstream, we can also use structure-based clustering to further diversify. We cannot do structure-based clustering. We cannot predict structures for the entire repertoire of millions of antibodies. But if we somehow pre-select a subset, then we can use structure prediction and structure-based clustering for further diversification and improvement of the lead panel that we're going to test in the wet lab. And when we apply clustering, the one thing that is worth looking is the distribution of the cluster sizes. So when we annotate antibodies, when we build clonotypes, we already fix sequencing and PCR errors, so we know that all the antibodies are reliable. And then when we cluster them, assuming that all antibodies are real, we don't have PCR or sequencing error, the size of the cluster tells us, let's say, the skewness of the repertoire and the diversity of the binders that we can have in this experiment. For example, here you see that for the just example, immune libraries, yeah, immune libraries from immunized animals, immune system already pre-selected some families. We have some discrete mid-sized clusters because there is already some pre-selection done by the immune system, yeah. And if we use synthetic libraries with no any pre-selection was done, yeah, before we enter the screening, we have we can have extreme dynamic range of the clusters, and there might be some really big clusters that can, dominate the entire round for sample. And the important thing here that the cluster size, how big the cluster of antibodies, is not equal to the signal of binding. So this is also important thing to know and not fall into a, we pick the biggest cluster. It means that this is antibodies that we want to include in our lead panel. That's not the case. The main signal, now go into the main signal, so which is the enrichment of antibodies or clusters of antibodies across the route. Basically, enrichment analysis gives us the main signal and the main data point for models to train for the affinity, or for selection, or for how to design libraries. That's why we're doing display panning. So the question here, how to identify binders by tracking the pathways of antibodies or families. Again, for the purpose of both lead selection and data foundation, the main recommendation that we have and that we learned from all the campaign that we ran, that to have a clear signal, we wanted, and to avoid this sampling of sequencing, we want to track enrichment of clusters. That's the reason why we did clustering. So we want to track clusters that are enriched across the routes, not individual antibodies. And we are annotating and labeling this data on the level of clusters. So what's the goal here? Basically, super straightforward. We just need to calculate the frequency of antibody in each of cluster of many antibodies in each of the rounds. Calculate the log-to-fold change of the abundance across the conditions, across the rounds. If we have negative control, which is very useful thing to have, and we always recommend to have negative control. And we saw so many experiments without negative control that just classify sticky binders. Use negative control to select really antigen specific, or sometimes we need B specific, or like depends on what we need, antibodies. So 2K metrics here that are useful and that are required for good data foundation. The log-to-fold change, the maximum log-to-fold change, the strongest signal round to round. So this is one metric that tells us whether this antibody was enriched ever across all of the rounds. If it was enriched with high max log-to-fold change, high means like 5, 6. This is high. The minimal that we're required to even classify the cluster is 2. This is one signal. This signal is not enough because sometimes, and I will show, we may have very different, let's say, profiles of how this max log-to-fold change looks like. The other signal is overall log-to-fold change, the net enrichment from the beginning to the end. Based on these two signals, the data label that is very useful to assign to cluster is what we call just enrichment quality, classification of enrichment quality. Stable binder is the one that has high maximum log-to-fold change and high overall. So it was constantly enriched monotonically through all of the rounds. Rescuer, and this is the biggest caveat that we saw. There might be antibodies that showed nice enrichment in some rounds, but then overall they have low enrichment. So they were enriched, but for example, on the last round, were overgrown by faster growers. If we look on the overall log-to-fold change, for example, only, then we will lose those rescuers, and they may be really good antibodies that are, not showing the overall enrichment because of the technical reasons. Weak binders, the ones that are enriched but not enough, and we also have parasites that are present in all of the rounds, but basically with no enrichment. And this label stable rescuer parasite, weak binder is a category that we feed to the whatever model we are using for affinity. Yeah, that's super useful and works nicely in our experience. And here are some examples of what you can expect from the enrichment profiles for different types of the experiments. Yeah, for example, this is immune library. 2 rounds of enrichment is enough, and we have a strong enrichment signal. So this is easy for AI. And then whatever type of library, whatever model we're using, this is like an ideal picture. We have strong signal, so it's clear. This is a synthetic example where you see now here we have some nice growers, yeah, that are growing across the rounds. But then we have some antibodies that will grow and then decline. Yeah, and there are many of this. And on the live part, I will show you more examples of this. This is really a pretty typical picture. So this classification into rescuer is still good, is important here. Yeah, sometimes if we under sequence, we may have pictures like this. You see here, we can have all good study level quality controls. There is a saturation, there is a nice enrichment or some enrichment on the gene index. But then when we look on the level of clusters, you see what we have. So there is a set picture of basically this is the picture where we can trust only this part. Only this part, yeah, where we saturated library to enough level, and we can rely on these 2 rounds to see some enrichment signals. So those are examples from the real experiments that we did and how it may look like. The very important thing is negative control, or in general control. Yeah, we may have a nice target enrichment, but for example, for phage display experiments, we may have sticky binders, antibodies that are actually enriched against anything. Yeah, and bind to anything. So if we are not using some sort of negative control, or negative control may be a fake target or a real target, but just a different one, or just a plastic. If we're not using negative control, then many of the antibodies that we classify as binders may be non-specific. Yeah, and this is one of the biggest caveats that we see if you're not running some sort of negative control. Yeah, this may really destroy the whole analysis result. And here you see the picture. So like here we are looking on the log to full change against target, and here log to full change against the negative control. This is a nice experiment. This is a very good picture because here this is, let's say, ideal scenario. Here we have a good set of highly enriched target specific clusters. So here they are, and this is a very good and strong signal. So based on this, we can cut everything below, and we can use those antibodies that are our target binders. And I will switch to a live example and show how this may look like on different types of experiments, what you can see. And if we classify our stable binders, our rescuers, if we remove non-specific antibodies, it's always useful to, yes, with our eyes, look on the data and quickly see whether now, with all this information, we're satisfied with the quality of the study. The useful instrument to see this, and by the way, AI can also classify this easily on the level of study. Yeah, what we're looking here, we're looking on the UMAP. So UMAP is basically a projection of the antibody sequence on the 2-dimensional space with the idea that 2 dots located closer to each other if they share similar properties. And if we visualize the entire campaign on this 2-dimensional visualization and let's say color antibodies by their antigen specificity and or filter for antigen specificity and color by the enrichment quality, we can see on the space that we have our stable binders here, here. So we have stable binders at different parts of the space. We have nice diversity. Like this is a good experiment. This is kind of an ideal picture. And also in this specific example, you see the overall cloud of antibodies with those small individual clusters. They are kind of distributed more or less evenly on the space. which means for us that the library, in terms of its underlying, let's say, structure, is more or less even. There are no biases, let's put it this way. And sometimes we can see this picture where we have a big cluster here and a big cluster here and a big cluster here and a small cluster here, which means that there is some global structure. Let's say we can use multiple different scaffolds for the synthetic library, or there is something else that worth looking in. And it depends on the experimental design that you're using. So before I switch to a live demo, just a quick note, the other very useful thing that we can do to feed this into a model, is simple developability and liability assessment. We can demand from the model to learn on the developability profiles, but one of the rules that is, let's say, one of the key rules for data foundation for machine learning or AI, provide as much structured data as possible. Yes, AI and machine learning models, they can learn developability and they can learn those sequence motifs from the data if you tell this is good and this is bad. But if you can, and it is cheap bioinformatically, assess for the sequence motifs that we know cause the amidation or fragmentation or whatever, like these ones, with high risk. If we know, we can quickly scan those sequence motifs in the data, structure them again, classify, for example, and we can classify those liabilities, sequence motifs into high risk and low risk, and feed this as a structured way, a structured data to the model. The model will thank you. We'll tell you thanks, because that will simplify the work. And this is also a very important thing to do if we want to train, to have a training set for both affinity and developability. This is obviously a cheap thing to add to the data to help with model training. The last point, obviously, is the real validation of the data. So, and this is the biggest bottleneck in any AI. So if we generate millions of antibodies with NGS, that's not enough. We have to do something, select a subset of those antibodies that we are going to test now as a ground truth, label them, for example, with their affinity or developability, measured in the wet lab, and then also add back to the NGS dataset, to the entire data that we generate with annotation, with clusters, with enrichment, and then form the final data set that we have for model training, which has the entire NGS picture, binders, non-binders, enriched and non-enriched, and the really validated sequences in the wet lab that are marked in that data set. This was validated and it is good. This was validated and it is bad. This was not validated. So with this, I will switch to a 5-minute live part to show how all this looks in practice, from the beginning, lead selection, integrating assay data, and then building the data layer for the AI. So let me switch to my user interface. Here I have a very interesting study. As an example, it's a real study with phage display experiment. There are 2 libraries, naive library and immunized library, multiple targets, and 3 rounds of phage enrichment. Now let's project what I was saying through all of the steps onto this real study. A very good and comprehensive study, a lot of data generated. Let's go to the quality checks and let's look on the rarefaction. So here you see the rarefaction curves and do you still see my screen? Yeah, I hope so. So here you see the rarefaction for all of the individual campaigns that were run in this study. And on the rarefaction, you see for all of the targets, we have more or less nice picture in terms of the rarefaction. So we have clear picture of saturation and to the last rounds, so the data is situated. So all is good here if we are not reading the colors. But if we read the colors, you can see that this green is round one, orange is round two, and blue is round three. So green here, orange here, blue here. This is good. This is bad. This is round one, this is round three, and this is round two. So something does not work well here on this plot. If we go deeper, if we go onto the Gini index and dominance, then we will see the picture that a really diverse set of patterns. So for some targets in this same library, this is the same starting library, and then just different targets and independent arms of phage panning. That's a public study. I think it's published in Nature last year. So very, very good paper. For some targets, we see a good enrichment signal. So you see there is a clear growth of the gene index. Some clones are expanding. And for some targets, the pattern is strange. So something didn't work here well. So we have a dip in round two, then we have something at round three. But basically, it means that in this specific experiment, there is more or less no enrichment signal. Here, all is good. This target is good. This target is good. This is just bad. Yeah, this is good. And you see how diverse with the same library, same experimental design, absolutely everything same. Yeah, just different targets, same lab, how different the picture is. Same sequencing machine, same depth of sequencing, but the picture is very different. So imagine if we use this data without, let's say, understanding. We can look for the binders in each of these data sets, and we will find enrichment signals. Even here, we will even here, if we look on the antibodies and their abundances, we will see that there are some antibodies that are growing. But overall, without study level label that this is flag, there is no good enrichment. We will mislead the model. So going deeper into enrichment analysis for good targets. Yeah, so this is one of the targets with good rarefaction, with a good gene index growth. And what we see for this specific target, we see the picture of enrichment, which is pretty different from what I was showing as ideal. So again, we have antibodies that showed a nice enrichment from round one to round two, and then declined downstream. So we classify such antibodies like this as a rescuer. Because yes, here clear, there was a real enrichment, a good enrichment, a significant enrichment, yeah? But then it went nearly down. Why? Should be something there. We cannot just drop it. So we keep it as a rescuer. For other targets, let me show you for other targets. The picture, more or less same. Again, we have rescuers and we have nice stable growers. And you see how it's different, round one, round two. So stable binder here is this one, for example. You see it's nicely enriched across all of the rounds. And these are our rescuers. If we look on the control in this specific experiment, this is a very deep biological study. So there was some control, which is not like a usual panning against plastic. So there is some complex biology behind, but just to show you a pattern. So this is a control scatter plot where the negative control is a actual real target. Sorry, a negative control is a real actual, protein. It's not plastic. We have a picture which is not easy to let's say accept without this picture, we would select a lot of the antibodies that are non-specific. So you see this is the enrichment here against the target. And you see that we have many antibodies that are enriched both against the target and against negative control. And the winners, the most enriched, and this one, the most enriched antibody, you see, it has the highest max log-to-fold change. It has the same, not the same, but also very high log-to-fold change against the control. And if we look for some other targets like this one, here the picture even worse. You see these are the winners in terms of the just enrichment. Those clusters are enriched a lot. Yeah, very good binding, if we assume that binding is proportional to enrichment, but they are all from the control. So control is important to select the true specific binders, maybe not that enriched, but specific to what we're looking for. Yeah, if we look on the space, yeah, basically this is what I was explaining on this uh, on this space. You can see structures. You can see structures. Yeah, so it's not very even. And the reason is the initial library. So the initial library has structures. Those structures may be different scaffolds, different lengths of the CDRs, how we design the library or how we use it, pooled animals. There are maybe different reasons, but we clearly see that this library has some global structure. I projected here enrichment quality against one of the targets. Yeah, and you see that our stable binders, they are distributed across distinct parts of this space, and they are located in one, let's say, in one global group of binders. That this group probably has some unique pattern inside this group, which we can further explore, of course. Yeah, what is specifically there, but it's a separate question. Yeah, it's also useful when we see some cluster of antibodies that is all of the antibodies that are enriched within the same cluster. It's always useful just to look with eyes on this cluster and see whether there are some specific motifs of the sequences there, etc., etc. But this, like for us, skin bags for AI, this picture is just enough. Let me go to the last point, to few last things. So when we're integrating the assay data, so here in this set we have a lot of antibodies that are really validated, and we can import those antibodies from the functional validation and map them back on the NGS and see how many of the NGS variants we have of the specific like antibody that we pick. Yeah, we have like a lot of them. So a lot of the validated antibodies have nice cousins and nice variants in our NGS data. We use this step only when we do functional validation. And let me show how we first select the antibodies for the validation. So we have all these data points. We have the enrichment, we have the clustering, we have developability, and by the way, just one thing. In this specific experiment, if we, let's say, filter our space to only antibodies that do not expose critical developability problem, then obviously the number of antibodies will be reduced significantly. So the amount of antibodies that do not expose developability problems is much less than we have in general in NGS. And this signal is very, very useful for any model. So when we select the panel, there is one thing that's super useful for AI and that we found is working nicely for whatever training we do. So when we set up experiment, we need to select some antibodies and validate them with a wet lab. We recommend two-step approach. The first step, select a large panel, 500 of antibodies. How we select? We filter for antigen specificity, we filter for the enrichment quality, stable binders and rescuers, we filter for the developability. In most cases, obviously, that's very, very useful. We use clusters to diversify, to pick antibodies from distinct clusters. Yeah, and then we prioritize them by the frequency. We want to select top antibodies within each cluster by its frequency in the last round, for example, or by the enrichment. Yeah, and developability cost. With this, we have a panel of 500 candidates or 1,000. That's number depends on you. Yeah, obviously we're not going to synthesize 500 of antibodies. Yeah, but we can use this 500 pre-selected antibodies, and we can see that on the space they are located at distinct parts, more or less. Still, there are some like clusters of similar antibodies there. Yeah, but we can use this 500 of antibodies to predict structure. Yeah, I didn't cover this on slides, but we are finding this recently more and more useful. So we can use this 500 antibodies to predict the structure with, let's say, immune builder. Immune builder is the best so far for two purposes. One purpose is to cluster based on the structure. Yeah, and you see from 500 antibodies, looking on the clusters with distinct structure, we have like just 6 clusters, much less than 500. Yeah. So this tells us that the diversity, the structural diversity is less. We can also use structures to better assess developability properties, looking on the surface. It's not the ground truth, but it helps us to rank antibodies. And then with this cluster and developability profiles based on the enrichment, we can select from this 500, let's say 96 or 20 or whatever number of leads based on the structure clusters, and developability cost now coming from the structure. Select this 95, 96 or whatever number of antibodies, synthesize, validate in the wet lab, import with assay data, and that's it. The data is ready. So with this, we have all the data. A big table. Obviously, under the hood, it's not a table. It's not Excel table. There's bioinformatics standard formats that's used, Parquet files, etc. But it's a big table, annotated with all the information, with all the properties, with the enrichment, with the study level metadata, with all the information. You see how many data points, how many signals we can include with this layer that is now usable for training the model. And that's basically what is minimum required for any meaningful training. Okay, data today, AI tomorrow. We need a clear data layer with all the information that we can bioinformatically collect from the data in systematically and reproducible way. Yeah, enriched with the functional validation that allows to build the good models specific for your data, for your experimental design, for your platform, for your libraries tomorrow. I am switching to questions. Monica?
Okay, great. Yes, so we have a few questions. One is from Fadir. Thank you for the nice talk. Quick question. Why do you choose a UMAP and not TSNE, for example, for sequence clustering / analysis?
It's a very good question. So we choose here UMAP. We try it. So we do both. Let's put it this way. Just for specifically antibodies, VHH, and display experiments, in our experience, UMAP gives better resolution and better ability to distinct different families, different features, etc. TSNE also works. It's a matter of preference.
Thank you. And then we have another question. I would like to understand the numbers involved in the binder workflow from target binding to sequence liability removal to final functional characterization. Do you get high confidence to down select 100 clones at the end for production with good diversity?
Exactly. That's what I was showing here. That's the obligatory part. So we need to select this 100 or 500 or whatever based on all those enrichment, developability. We select this. There is no AI on its own. We need to select 100 or 500. Here I gave an example how we do this. We select, let's say, a larger set, then we model structure and use the structures to refine and get the final 96 yeah, sometimes less, candidates that we are confident good for synthesis and validation, we may be wrong. That's the only way is to try. The better we can characterize, the more we can characterize, the better. If we can do this for 1,000, it's better. So that's limited to the capacity. Yeah, and we're pretty confident in those 96. And then with all this information, we can improve, for example, the library design. So let's say we downstream will have a hit rate. How many of those 96 are good based on the signals that we used here to select? Yeah, we in those signals we can include that's where AI shines. We can include a model, a machine learning model that takes into account all the enrichment signals and other signals, and let's say mutations and mutations in different positions, and tell us these 96 are better than the ones that you select. But basically, yes, this is a required step.
Great. And how much data do we actually need before an ML model gives useful predictions? Do we need thousands of sequences or millions?
Today, millions. If we just start with from scratch, we need to have millions. Once we improve the model, and we can use, for example, a model for. Oh, sorry, I stopped sharing. We can use model for rational library design. We can design libraries of less diversity with less number of antibodies, and then we will have less size of the set, obviously. But this requires the foundational. So it's progressing. You start with the bigger data sets, and then you refine model. You need less data to fine tune those models.
Okay, so that's all the time that we have for questions. Stan, do you want to close up?
Yeah, well, thank you very much, everyone, to joining this meeting. I hope it was useful. And we have all the algorithms and all of the ideas that I was talking about as an open white box environment Platforma. Many of the algorithms are available on the GitHub, and many of the parameters that we use are available on the GitHub. Yeah, you can run Platforma on your own. It's free for academic research, and for industry, feel free to reach us and request a demo. Thank you.
Thank you so much and have a wonderful